The goals of this protocol are to describe steps to reconstruct the morphology of virus-labeled neurons and to perform independent, unbiased cluster analyses on populations of labeled neurons to enable comprehensive characterization of morphological metrics along distinct neuronal subclasses.
We outline a novel approach for collecting and analyzing neuroanatomical data to facilitate more comprehensive sampling and unbiased classification of morphologically unique neuronal types within a selective neuronal population. The main advantage of this technique is that it enables large-scale analysis of neuronal morphology and utilizes unbiased methods to classify distinct neuronal subclasses.
The most challenging aspects of morphological reconstructions are selecting well-isolated neurons to reconstruct, assigning the proper Z-depth, and aligning arbors to continue reconstructions across adjacent tissue sections.
Begin a reconstruction by selecting a slide bearing a stained 50-micron brain tissue section from a primate stereotaxically injected with modified rabies virus expressing EGFP. Place the slide in the microscope slide holder and focus on a single section of interest using a low magnification objective. Make sure the camera image is visible in the live image by properly positioning the camera shutter. Choose a reasonably isolated labeled neuron within the brain structure of interest to enable unambiguous reconstruction of dendritic arborization. Switch